Hi, my name is Yi Bao. I'm one of the lead authors of an article published in the Journal of Clinical Investigation titled CDK12-13 Inactivation Triggers STING-mediated Anti-Tumor Immunity in Preclinical Models. This is a follow-up study based on our previous observation that CDK12 inactivation increases T-cell infiltration in both human and mouse tumors. In this study, we aimed to define the mechanisms underlying these phenotypes and to elucidate their therapeutic implications. With the hypothesis that CDK12-13 represents a therapeutic target for inducing anti-tumor immune responses and enhancing the efficacy of immunotherapies. At a glance, this study demonstrates that while intact CDK12 and 13 ensure proper transcriptional processing, their degradation induces transcription replication conflicts and R-loop accumulation, leading to cytosolic DNA release and subsequent activation of the CGAS STING signaling pathway, which has been reported as an important pathway that activates anti-tumor immunity and enhances response to immunotherapies. We began by performing spatial transcriptomic analysis, which showed STING activation enrichment in CDK12 inactivated tumors compared to the wild type. We further validated this using bulk RNA sequencing data, which revealed that low expression of CDK12 and 13 was associated with higher expression of STING signature in many different cancer types. And importantly, low expression of CDK12 and 13 was also associated with improved response and survival in cohorts treated with immunotherapies. We next tested whether inactivation of CDK12-13 could trigger STING activation using the selective CDK12-13 degrader YJ1206 and gene-specific knockdown approaches. We found that targeting CDK12 and 13 activated STING signaling across multiple preclinical models in vitro. To determine how STING signaling was activated, we discovered that CDK12 and 13 inactivation led to transcription replication conflicts and R-loop accumulation, which subsequently caused the release of cytosolic DNA that activated STING through CGAS. We next evaluated the efficacy of CDK12-13 inactivation in animal models and found that it suppressed tumor growth and enhanced the response to immunotherapy. As expected, CDK12-13 inactivation also triggered STING activation in tumors in animal models and increased effector T-cell infiltration in tumors. Importantly, through gene knockout and CD8 T-cell depletion experiments, we demonstrate that both STING signaling and effector T-cells are essential for the efficacy of CDK12-13 inactivation. In summary, targeting CDK12 and 13 induces transcription replication conflicts and R-loop accumulation, leading to cytosolic DNA release and subsequent activation of the CGAS STING pathway, which drives anti-tumor immunity. In conclusion, this study identifies STING activation as a key driver of T-cell infiltration and establishment of an immune-hot tumor microenvironment in CDK12 mutant cancers. We further demonstrate that CDK12-13 inhibition or degradation activates anti-tumor immunity and improves response to immunotherapies. This study provides strong rationale for clinical trials evaluating CDK12 and 13 antagonism in combination with immunotherapies.